I'm delighted to introduce our next speaker, Dr. Joan Mannick, who will continue with the topic of gero modulators and pharma medication. Sorry, more?
So before I start, I just want to give a shout out to Eva, who probably has done more to catalyze the whole field of longevity medicine than anybody. So really amazing. So I'm going to talk today about our company, Tornado Therapeutics, and give you an update on the progress we've made. So Tornado is developing best-in-class next-generation rapalogs that I think we'll be able to get to you in the next few years. This isn't something a long way off. So rapalogs are derivatives of this oral mTOR inhibitor called rapamycin that you've heard a little bit about today. And they're FDA approved for the treatment of many diseases, including cancer. But what's exciting about rapalogs for our field is they have additional untapped therapeutic potential because this one drug, rapamycin, is the best validated therapeutic to extend lifespan and health span in model organisms. And they live longer not just because they have less cancer, they have a delay in incidence of multiple aging-related diseases. So this sort of gets to a similar phenotype to what Nir was talking about with metformin. So it's likely, given all this preclinical data, that they are going to have benefits for human aging. So we've in-licensed a portfolio of next-generation rapalogs from Novartis that are predicted to be best-in-class compounds with better safety and/or efficacy as compared to currently available rapalogs like rapamycin. Our lead program is going to enter the clinic in the first half of next year, and we're going to start with 2 indications that are de-risked not just by preclinical data, but actually by clinical data from other mTOR inhibitor programs. The first is oncology, and the second is actually a geroscience indication. We're going to try to improve the function of the aging immune system to decrease the severity of viral respiratory tract infections. So mTOR is a protein kinase, and it's the key protein that links the availability of nutrients to growth and reproduction. So during periods of feeding, mTOR is activated, and this upregulates growth pathways. Then during periods of fasting, mTOR is inhibited, and this upregulates stress resistance and repair pathways. And these pathways that are upregulated during fasting are not only important for surviving periods of famine, they're also linked to lifespan extension. And this may be one of the reasons that there are health benefits of intermittent fasting. It's because it's inhibiting mTOR. So the problem as we get older is mTOR may become dysregulated. So this is a figure just showing in young mice, you can measure mTOR by measuring the levels of a phosphorylated protein called S6. So you can see in young mice when they're fed, this phosphorylation of S6 increases, mTOR is activated, and this increases protein and nucleotide and lipid synthesis so organisms can grow and reproduce. And then during periods of fasting, mTOR is inhibited and the protective pathways are upregulated. So mTOR should sort of have this circadian rhythm on during the day when you're eating, off during the night when you're fasting. But there's now data in multiple human tissues and mouse tissues that mTOR gets hyperactive with age. And David Sabatini published an interesting paper in Nature showing that in old mice, at least in the liver, mTOR no longer responds to fasting. It just stays on all the time. So there's no periods of the day when mTOR is inhibited and these protective pathways are upregulated. And this suggests that just turning mTOR down to young levels or turning it down intermittently may have health benefits. So how can we turn mTOR down in humans? Well, this gets to rapamycin, which has an interesting backstory. There's a water in a crater on a remote island in the Pacific Ocean called Easter Island, or also known as Rapa Nui. And it was reported by local residents that it had some sort of health-promoting benefits. And some investigators studied soil samples around this volcanic crater and found that it contained a bacteria that produced a molecule that was a very potent and specific inhibitor of mTOR, and they named it rapamycin after Rapa Nui. So it turns out that inhibiting mTOR with rapamycin or genetically extends lifespan in every species studied to date: yeast, worms, flies, mice. And these organisms span 1 billion years of evolution, so it's likely that the health span and lifespan extending benefits of rapamycin and its derivatives are going to also extend to humans. So the validation for this pathway and for this one molecule is really impressive. In all of these organisms, multiple labs have been able to reproduce the same finding of lifespan extending benefits. And in the study on the left, which was a famous study done by the NIA Interventions Testing Program, even when rapamycin is given late in life to mice at the age of a 60-year-old human, it is sufficient to extend lifespan. And then Dudley Lamming did an interesting study on the right showing even if you give rapamycin just once every 5 days in older mice, that also is sufficient to extend lifespan. So given all of this validation, given the fact rapalogs are approved for use in humans, why haven't we done more to study whether they have benefits for aging-related diseases? And the first problem is that the currently available rapalogs, including rapamycin, have limited or no remaining patent life, and this has diminished investor interest in pursuing new indications because it's very costly to pursue new indications. So Novartis realized this back in 2014 and put together a large team with their expert rapalog chemists to develop new rapalogs that could be improved on the first generation and would have the patent runway to develop for aging-related diseases. This was a really tough task because so many rapalog structures have already been disclosed in previous patent filings. But after many years, they came up with a portfolio of really interesting compounds with patent runways to 2038 and beyond that can be developed for aging-related conditions. But by the time they developed all these compounds, Novartis had a change of management. They decided they weren't going to focus on aging biology anymore, and so this program became orphaned. But we were able to bring it into Tornado Therapeutics to keep it moving for aging-related conditions. The second problem that has hampered moving rapalogs forward for aging-related conditions is safety. So rapalogs are approved at high doses that completely turn off mTOR, and they have a lot of side effects. But what's exciting about this portfolio is some of the compounds are predicted to be best in class with improved safety, as I'll show you, compared to first-generation rapalogs, and that gives us even better opportunity to move them forward for aging. So if you had to design a rapalog that would be best suited for targeting aging biology, what would it look like? Well, mTOR signals in two multi-protein complexes. One's called TORC1 and the other's called TORC2. It's inhibition of mTOR in the TORC1 complex that's responsible for the lifespan extending benefits of rapalogs and mTOR inhibition. Inhibition of mTOR in the TORC2 complex has actually been shown to decrease lifespan and cause hyperlipidemia and hyperglycemia. So an ideal rapalog for extending lifespan would be predicted to be one that selectively inhibited mTOR in the TORC1 complex. So the currently approved first-generation rapalogs like rapamycin potently inhibit mTOR in the TORC1 complex, but also partially inhibit mTOR in the TORC2 complex. We've now synthesized and characterized the majority of the compounds in the portfolio we've in-licensed. And what's exciting is the majority of compounds in our portfolio are TORC1 specific rapalogs. So this is a geroscience gold mine, and we're really excited about moving these compounds forward for aging-related conditions. Then 26% of the compounds have TORC1, TORC2 specificity similar to rapamycin, And 14% are what Novartis called super rapalogs. They're even more potent TORC1 and TORC2 inhibitors than first-generation rapalogs, and we have some data that these may be quite effective for autoimmune diseases. So this is just some data looking at the relative TORC1 and TORC2 inhibitory activity of our compounds versus everolimus on the left. So you can see everolimus has a sub-nanomolar IC50 for inhibiting TORC1, and it also partially inhibits TORC2. Our lead compound, TOR101, also has a sub-nanomolar potency for inhibiting TORC1, but it doesn't inhibit TORC2 at all, even up to 300 nanomolar, which is the highest we've tested, and is 1,000-fold higher than the TORC1 IC50. TOR103 is a very similar compound in the portfolio that's TORC1 specific, and TOR104 is a super rapalog that's a more potent TORC1, TORC2 inhibitor than everolimus. So I'm going to focus the rest of the talk on this lead compound, TOR101. So just looking in rats dosed with TOR101 at 3, 10, or 30 mg per kg, we look at TORC1 activity, again, by measuring the levels of phospho-S6 in liver, kidney, and muscle at 3 and 24 hours after dosing. And you can see the lowest dose we tested, 3 milligrams per kilogram, led to almost complete TORC1 inhibition at 3 hours, and then there's a dose-dependent recovery at 24 hours. So these compounds are behaving like they should. They're inhibiting TORC1 in vivo. What's most exciting is the preliminary safety data. So before you go into the clinic, you have to do what's called GLP toxicology studies in 2 species to find the lowest dose that has no adverse effects. So for everolimus, this is data from Novartis, when rats and non-human primates are dosed for 28 days, the lowest dose with no adverse findings is 0.25 mg per kg for rat and less than 1.5 mg per kg for non-human primates. So we have finished our 28-day GLP toxicology studies and are still waiting for the histopath, but we have histopath back from 14 days of dosing, and our preclinical toxicologists have said this will predict what you're probably going to see after 28 days of dosing. It'll be somewhat predictive. So what has surprised everyone is we're seeing no toxicity up to the highest dose we've tested, 250 mg per kg in both rats and non-human primates. Now, we're likely to see more tox as we get dosing through 28 days, but this is really surprising and suggests that these TORC1 specific compounds are going to be safer than the TORC1-2 previous generation of rapalogs. The most exciting data is we finally got some data back from our rat GLP toxicology studies where we can look at lab abnormalities induced by 28 days of dosing of everolimus and lab abnormalities induced by 28 days of this TORC1 rapalog, TOR101. So there's certain lab abnormalities that occur in both humans and rats that are dosed with rapalogs. And those include decreased lymphocyte counts, decreased platelet counts, and increased lipids. And you can see these are seen at really low doses of everolimus in rats after 28 days of dosing, in general less than 1 mg per kg. So here's what we've seen with TOR101. We're seeing no decrease in lymphocyte counts, even up to 250 mg per kg. We're just starting to see some decrease in platelets, but not until 250 mg per kg in males. There's a dip at 50 mg per kg in females, but then it goes back up at 250 mg per kg with a higher exposure. So it's not even clear this will cause thrombocytopenia in females. There's no increase in triglycerides. And in the females, there's no increase in cholesterol to 50 mg per kg, which is 100 times higher dose than where you see it with females with everolimus. Where we don't know if we have a differentiation is increased cholesterol in males, because our lowest dose, 10 mg per kg, there was some increased cholesterol, so we have to move the dose farther down to see how it differentiates from everolimus. But this is really exciting because it suggests TORC1 specific rapalogs are going to decrease the risk of hypertriglyceridemia and cytopenias which are a big problem with first-generation rapalogs and limit how high you can dose them for aging-related conditions. This is also very exciting for indications like oncology, where that decrease in blood cell counts caused by rapalogs limits how they can be used in combination with other oncology agents. So there are so many different indications for which rapalogs have been shown to have benefit either preclinically or in the clinic. And we've decided to focus on 2 indications where there's the most placebo-controlled data that our compounds should have benefit based on previous generation of compounds. And that, again, is oncology and viral respiratory tract infections. I'll just talk a little bit about those 2. So one of the interesting oncology indications for next-generation rapalogs is a cancer called diffuse large B-cell lymphoma. So the standard of care is a chemotherapy regimen called R-CHOP. And you can see in the red lines on these 2 graphs that the event-free survival is about 30% with R-CHOP, 30% of patients don't respond and relapse. And the overall survival is pretty good. After about 2 years, the overall survival is close to 90%, and this is in a good prognosis population in these graphs. So since you can't get better than a 70% response rate, Mayo did a study where they added our first-generation rapalog called everolimus to R-CHOP to see if it would improve response rates. This was a small trial, but the efficacy was remarkable. So the event-free survival when you add everolimus to R-CHOP was 96%. Only 1 event occurred, and it wasn't even a relapse of the cancer. It was a new, different kind of cancer that occurred, a follicular lymphoma. The overall survival was 100%. This was published in 2017, and as of 2023, not a single of these patients has relapsed, and everybody has survived. So basically, 100% complete remission with a combination of everolimus plus R-CHOP. This was really exciting. ECOG and the NCI were wanting to do a phase 3 trial of everolimus plus R-CHOP versus R-CHOP alone, but it couldn't move forward because everolimus was coming off patent. But this provides a really interesting opportunity for our next generation rapalogs. So we did a study of our lead compound TOR-101 plus R-CHOP in a DLBCL model, just to make sure we could reproduce that efficacy that was seen in the clinic with everolimus. So you can see in black the tumor growth with vehicle, in green tumor growth with R-CHOP. This is a relatively treatment-resistant model. In blue, tumor growth with TOR-101, and in red, tumor growth with a combination of TOR-101 plus R-CHOP. So this is exciting because it looks like it's recapitulating the efficacy seen in the clinic with everolimus. So that's an indication we're excited about moving forward in. And then the second indication is more near and dear to this group because it's a geroscience indication. So we all know COVID-19 is more severe in older adults, but it turns out almost every viral respiratory tract infection is more severe in older adults. So this is data from the CDC that was published in New England Journal before the COVID-19 pandemic, just looking at rates of hospitalization in people who get a common coronavirus respiratory tract infection, an influenza virus infection, or a rhinovirus infection. And you can see with increasing age, these respiratory viruses cause increasing rates of hospitalization. So the way we currently deal with this is one drug, one bug. We get a drug like Paxlovid that targets one kind of virus, but the problem is when you target a virus, the virus develops escape mutations and viral resistance, and older adults are now susceptible to all the other hundreds of respiratory viruses. So we're saying, what if we target the dysfunction of the aging immune system, particularly how the aging immune system responds to viruses, which is defective? This would protect them from all sorts of different respiratory tract infections without inducing viral resistance. So we spent a lot of time with a previous mTOR inhibitor compound, seeing if we could improve antiviral immunity in older adults with mTOR inhibitors. And I've talked about these data a lot, and I'm just going to talk about the lessons learned, which was low doses of mTOR inhibitors are really well tolerated in older adults. 2, they always upregulate, in every study we've looked at, interferon-induced antiviral immunity, which is deficient in older adults and one of the reasons why they get more severe infections. 3, in our previous trials, we were probably using the wrong endpoint. We always see a bigger effect on mTOR inhibitors decreasing the severity than the incidence of viral respiratory tract infections. 4, they may have viral-specific effects. There seem to be some viruses that have a bigger treatment effect than others. And last, they seem to have the greatest benefit in people 75 and older. And it may be lots of people 65 to 75 kind of are okay, have a relatively normal antiviral response, and it isn't until you get older that there's a more homogeneous defect. So given these lessons learned, we did a trial that was small just to see could we use an mTOR inhibitor to decrease the severity of one kind of viral infection, COVID-19, in very old people, residents of nursing homes experiencing a COVID-19 outbreak. There were just 18 people who were treated with an mTOR inhibitor called BEC, 17 people treated with placebo. When a nursing home had a COVID-19 outbreak, the people who didn't have COVID were randomized to the mTOR inhibitor or placebo, treated for 4 weeks, and then followed. This was before there were any vaccines available, and 23% in this placebo group got severe COVID-19, and half of them died, and no one in the mTOR inhibitor treatment group even got a symptom of COVID-19. Small trial has to get reproduced in a bigger trial, but is giving us some more confidence that we're starting to hone in on the right clinical development path for this indication. So just here's the pipeline. We're just completing our IND-enabling studies with our lead compounds, so we'll get into the clinic early next year. We have a backup compound that's less expensive to make, which has commercial advantages behind it. We have a compound that's a super wrap log for autoimmune disease, and then another set of compounds for dermatology indications. And I'll stop there. Sure.
As always, Dr. Mannick. I'm sure there's plenty of questions. We will take one. Maybe you can say loud, and then I will repeat.
Okay, thank you so much. Super exciting. I actually did a PhD on rapalogs and rapamycin engineering 20 years ago, and then the program was bought by Wyeth and then the market, so maybe some of my work is...
Absolutely.
But anyway, my question, I was really surprised by seeing that mTOR is no longer inhibited in elderly mice, even by fasting. So do we have any data on healthy aged individuals, so not obese, so healthy aged when doing fasting, do we have any data on either transcription or proteomics and so on mTOR?
Right,
so
no.
Mice, but
what I can say is we don't know, or I don't know, data where fasting in older, particularly like 75 and older, is it actually inhibiting mTOR? I love this because I don't like to fast, so I just say I'm too old, it's probably not going to work for me. But we don't know, we need to look, yeah.
Thank you so much. Great, thank you so much once again, Dr. Joan Mannick. And I'm